So in the last lecture, we were talking about 16SrRNA sequencing, right? So in continuation with that, some other aspects, more aspects about 16SrRNA we have to discuss. And in addition to that, 18SrRNA. So when we talk about 16SrRNA, we know that it is used for taxonomic purpose, right? And when it comes to bacterial species identification via 16SrRNA gene sequencing, there is no universal definition to talk about if there is 99% similarity, 65% similarity, or 89% similarity would be a guide for or criteria for establishing a species match, right? Different authors have framed different definitions for that. If none of these studies does the definition of species match ever exceed 99%, so similarity is 99% and diversion is less than 1%. When it comes to ribosomal units, right? So SS unit, small subunit we are talking about. If that data has been compared to identification results, then you are going to like use different ways to do it. Conventional methods or commercial methods are there. We'll be talking about whenever the reference comes. So whenever these studies were carried out, general observations are made, like say higher percentage of species identification is obtained using small subunit sequence that results than with the either conventional or commercial method. And most studies have found that 16S yielded species identification rates of 62 to 91%. So the closest match is microsec 500 database. There are different databases available. Commercial methods are there, conventional ones are there. So when it comes to identification using the specific database, using conventional method or using the uh, uh, commercial method, variations are there. But when it comes to example, say microsec sequence, a 500 base database sequence, then identification you talk about, it gives the closest match, right? For bacteria, they are difficult to grow or identify. The identification rates we have talked about unculturable bacteria, right? So even when it comes to unculturable bacteria, the database itself is not that rich. That is one thing. Secondly, there is difficulty in growing these organisms and identification of these organisms. When it comes to use of 16SrRNA identification for unculturable bacteria, sequencing gives you 62 to 83% than the values which are traditionally acceptable. Traditionally, what we say, more than 90% similarity shall be considered as the criteria for finding the species match or genus match, respectively. But when it comes to unculturable, the matches are between 62 to 83%. So how to utilize this data in classifying or taxonomic positioning of the bacteria? That is a question, right? Also, the problem is there when it comes to use the complete and accurate database and groups that are not easily distinguishable by 16S RNA gene sequencing. Which database to choose, whether to go for complete genome sequencing or to go for specific database for specific type of organisms. All these needs a kind of uh, trial and error. Now, when we talk about 16S rRNA in bacteria, 18S rRNA, it's parallel for eukaryotes. We know. So, small eukaryotic ribosomal subunits, that is what is 40S, right? So, 40S and 68, they constitute eukaryotic ribosome, and that is also is of use when it comes to identification. So, we have to talk about um, how and what are the how to go about it and what are the limitations. In the previous lecture we have talked about the methodology in detail. Now here are some kind, this lecture is like the comments on the 16 is RRN and 18 is RRN as a tool. So when we talk about the sequencing, here are general what we say terms, you can say. So high throughput sequencing, we know Illumina or microsec, just now we talked about it. So these are the things which are the high throughput sequencing technologies. These are going to read, and whatever the sequence you get is called as sequence read. Okay, the sequence read can be shorter, sequence read can be longer. So longer read sequence, when we talk, they are about 1,000 base pairs. And one of the technology which is used is 454 Roche technology. We have talked about it in the previous lectures. The concern is this is going to be a method which is cost intensive, so limitations are there when I talk about R54 technology. But yes, it gives you long sequence reads, about 1,000 base pairs. And another important factor when it comes to 16S rRNA, we have seen that is hypervariable regions and variable regions in the gene, right? So increasing the number of microorganisms and can be identified at species level is possible when you are thinking about long read lengths, and the technology used is 454. Now, against longer reads, we have short read length sequences also, which are going to span only the hypervariable regions. And the question is, are they able to resolve the taxonomic assignment properly? Right. So, the optimal community clustering confirmed with sequence reads of this length is an important advance in amplicon design. Now the thing is, what does it mean? Ki whenever you are talking about long read or short read, the concern is what amplicon you are going to design, and that is going to be very important, because specificity is based on the amplicon, and then the PCR product, and then analysis would go. So when it comes to compatibility of the fragment length, you have to choose a right amplicon size, and a right sequencing platform. A right sequencing platform, as in 454 technology is one, Millisec is one, or Illumina is one. These are the different platforms, right? So what you choose matters a lot. Now, okay, we will go to 16S first, and then we'll go to 18S. Now, this is prokaryotic ribosome. It is ATS ribosome, that is eukaryotes. So, this is small unit and large unit, small unit and large unit, as you are seeing in the diagram. And these are the components, that is 23S and 5S rRNAs in the larger subunit of prokaryotes, and 28S, 5.8S, and 5S rRNA in the larger subunit. But if you see the lower unit, subunit, then for prokaryotes you have 16S rRNA, and in 40S, that is lower subunit of ribosome, in eukaryotes you have 18S rRNA, right? So when we talk about ribosomal smaller parts of the prokaryotic ribosome, whether it is prokaryote, we have 16S, and in eukaryotes you have the comparable one, that is 18S. Now functionally they are constant. So it shows a mosaic structure of conserved and more variable regions. Mosaic is every time you will get variation in the pattern. So when it comes to the structure of conserved sequences, you will find a variation in that, and it is going to be important in intragenomic heterogeneity studies. So you will find that multiple 16S rRNA operons and is present in a very high copy number in all the cell extracts. So it is widely used as a target molecule in genosensor articles, or in finding out the sequence, and then finding out the taxonomic position of the organism. Now when it comes to the general methodology, whether it is 16S or 18S, what we are talking about, first you will have to extract the genomic DNA, then you are going to go for PCR amplification. You are going to have multiple copies of the desired sequence. Then you are going to go for uh, the regions which are ITS regions. In case of fungi, we will come to those, right? Those sequences are shown here, ITS 1, ITS 2, like that. And these are the most important regions when it comes to sequencing of fungi or eukaryotes. Now these sequences are sequenced by Sanger sequencing method, right? And then sequence blast is done, which is done with the help of universal or international sequence databases. They are used to find out what is the sequence present in these regions, and then search match is going to be there, and then you are going to uh, use search markers for phylogenetic analysis. So DNA you have extracted, you have taken the amplicon, you have amplified it, you have sequenced it using say Sanger's method, and then you have blasted the sequence to find the match using the databases like INSD, and then you are going for search markers for phylogenetic analysis. So of course, these search markers are available in international databases like Gene Bank, EMBL, or DOBG. These are the names which are providing you the reference sequence loci, which can be used for phylogenetic positioning of the organism. And here you are going to carry out multiple sequence alignments, and then ambiguities are removed, and you get to know how far is the percent similarity and to which group the isolate would probably belong to. So this is how the flowchart of the you can say the identification using molecular phylogenetic analysis methodologies. Right now, next comes, see, this is what is schematic easy way to represent the same thing. So this is little difficult to draw in the exam. So maybe we can use this flowchart. Right now, it is again same how you are going to isolate, how you are going to amplify, and the sequencing it and alignment of sequences, and then taxonomic classification using existing reference databases. So now when it comes to the 16S RNA set of primers for amplification, what we have taken into consideration that this there are particular sequences within 16S and same as in within 18S, and they are variable and hyper variable regions. In the previous lecture we have talked about V1 to V9, right? So here comes uh, the step ahead, choosing different set. You are not taking the entire set of V1 to V9, but we are talking here about choosing different sets of primers for amplification of different hypervariable regions. Maybe someone is choosing V1 to V3, someone is choosing V4 to V5, like that. So these are different sets they are chosen, and they are amplified only. So, uh, if you choose these different zones, amplification, and that is sequenced, and then that is used for finding the sequence match, surely you are going to have abundance of hypothetical bacteria will get identified, right? For example, in this figure, the species abundance distribution obtained. Now, if you are choosing V1 to V3 region, that sequence is amplified, sequenced, and sequence match is done. How many species are you going to see here is A, B, and C. The abundance, that is species abundance when you talk about here using the primer set 1 is going to show you some pattern like this. And if I choose V4 and V5 region, what kind of similarity pattern are you getting? Are you getting it the same? Surely not, right? So what is important is you have to go for a selection of an appropriate primer or amplicon that should be amplified. So using universal primer set, which is typically to amplify a great coverage of bacteria and archaea, whether it is skin sample for this particular example we are taking, but for any sample you have to go for specific primer that would give you a proper uh, 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 picture of the abundance, right? So this is just to show you that these are the different variable regions, hyper variable regions, and when you choose specific sets of these hyper variable regions, what is going to be the outcome? Now, when I talk about sequencing, as we have discussed earlier, Sanger sequencing or next generation sequencing or metagenomic or whole genome shotgun sequencing, all these techniques we have learned individually in different lectures. So, when it comes to Sanger sequencing, uh, it is going to be a method which is going to require PCR amplified or clone target and incorporates radioactive fluorescent dideoxynucleotide. We know that. In next generation sequencing, as we just spoke about, 454 or Illumina or solid platform or pyro sequencing is used. Similarly, you have metagenomic This term describes the use of cultivation independent genomic based investigations. So unculturable bacteria, for example, where which are, I mean, you are not going to have a pure isolate, but you are going for a natural sample, say water sample or soil sample, and from that you are going to proceed with an uh, metagenomic, that is fundamental phylotyping of the bacteria that you are going to do, and this is a cultivation independent method to begin with, right? So of course details of metagenomics we are going to study in next lectures, but this is another way to approach 16s rRNA sequencing. And then next comes the whole genome shotgun sequencing. That is not the mm, uh, limited to 16s region, it is for entire DNA. Now these different methods are going to be applicable, and any of these methods maybe 800,000 base pair sequence when you are reading, then Sanger sequencing method is more than enough, right? So now the concern is, whatever the method that you use for 16s RNA sequencing, what are the issues, right? Now sequencing results are generally only semi-quantitative. Sequencing cannot distinguish between living or dead bacteria, and some phyllophytes cannot be differentiated at species level. So these are some of the limitations of 16S. When it comes to clinical microbiology or identification of pathogenic bacteria, then the clinical relevance of the microorganism with its genus and species level identification, that needs to be done, right? So the challenge in identifying is that the definition of bacterial species has traditionally been on phenotypic characteristics. So, matching of these two methods to give you a right identification is a limitation or challenge, you can say. It is not that that always 100% going to work together. So, this raises the issue of defining the species. Additional issues relating to the bioinformatics that is required to store and process vast amount of sequence data. You cannot just have only a limited number of reference data and you are trying to match with it. If it falls with similarity to it, or uh, no similarity with, you conclude based on that. That cannot be done. You have to have a vast store of data, right? The sequence data which is going to be used as reference data. The need for chimera checking. What is chimera? You know, these are the hybrids which get generated, hybrid amplicons, right, get generated during PCR. Of course, the probability is very less, but yes, that is required to be taken care of, and then only you can expect accuracy in identifying the particular isolate using the 16S rGNA, rDNA or rRNA gene database. Now another point is there, existing and ongoing development of analysis tools that assist in phylotyping of 16SrRNA gene sequence include but are not limited to. So what are these phylotyping tools which are used? They include Unifrac, Mothur, QIIME. These are the names, right? So we have talked about this methods which are used for sequencing, right? So Sanger's method, then we have talked about shotgun method, then we have talked about next generation sequencing, Illumina, etc. Similarly, here we are talking about the databases which are commonly used, and they are Unifrac Mothur, Ribosomal Database Project Database, and QIIME. So of course, there are challenges, and the challenges that we discussed till now. The use of 16S rGNA based methods have already increased our understanding of diversity of organisms, may it be skin organisms or any pathogenic organism, right? Or may it be any non-pathogenic and soil bacteria, whatever it may be. So when it comes to pathogenic bacteria, you have specific data sets. When it comes to non-pathogenic ones, soil-based ones, based on their niche, the reference database should be used, and it should be giving you adequate similarity profiling. Now here, the concern that we were talking about, hypervariable regions. So there are many studies that target different regions of the 16S rRNA gene. So V3 to V5, V1 to V2, like that up to V8 to V9. Now the concern is, if you take V6 region, is not optimal for sequence analysis that are directed for taxonomic assignment and community cluster. But the sequence read between V2 and V3 regions is good for community clustering and taxonomic assignment. Right? The most informative 16SrRNA region to amplify may also depend on analyzed environment. That is very important. So a study for diagnosis of pathogenic bacteria, for example, we talk about, it has showed that in a mixture of 110 different bacterial species, including common bloodborne pathogens, CDC defined agents, and environmental microflora. So, what you are talking about? You are talking about pathogens which come from blood, bloodborne pathogens, and 110 different bacterial species were there. What was found that V2 and V3 regions are more suitable for distinguishing all bacteria species to the genus level except for closely related Enterobacteriaceae. So whether it would be V1, V2, V3, V5 or V8 or V9, which hypervariable region would be giving you a sequence that would give you correct identification or closest possible correct identification is going to differ and is going to depend on the environment that is analyzed, the amplicon that is used, and the reference database that you have. So this is just to share one example. Ki it's not the story that 16S RNA sequence is done and the results are obtained and you are going to get classification to the taxonomic level and clustering also 100% done. No, not like that. There are certain limitations or challenges and one has to answer those as well. Now next comes then is it, what is the use, you know, what degree of utility you have of 16SrRNA? Then it is like we know it is highly useful in regards to bacterial classification. It has low phylogenetic power at species level and poor discriminatory power for some genera. That are the limitations of 16SrRNA. And when you talk about DNA relatedness, studies are necessary to provide absolute resolution. So analytical environment, the reference database, the type of amplicon, all these things are the limitations. But in addition to that, what is said is it is not really very good at resolving the isolates at the general level, right? And you have to go for DNA relatedness, that is hybridization tools. They are necessary for taxonomic uh, profiling of the organism. Now, example is there. For example, Bacillus. Now, type strain of Bacillus is Globisporus and Sacrophilus share 99.5% sequence similarity when they are scanned for 16S rRNA gene. But the DNA level exhibit only 23 to 50% relatedness in hybridization studies. Are you getting the question? The concern is 16S rRNA is showing about more than 99.5% similarity, but when DNA hybridization studies are done, same isolates are showing very less relatedness, as less as 23 to 50% relatedness. Right? Then the question is what data is to be used for uh, taxonomic positioning of these organisms? Are they bacillus species only? Is a question, right? So what we expect, okay, bacillus, and this is also bacillus, so good number, that is 99.5% similarity when it comes to 16SrRNA. But at the same time, the same organisms, when analyzed for their DNA level similarity, it is very less, 23 to 50% only. Now, another example is type strain of Adversiella. They exhibit about 99.35 to 99.81% similarity, means almost very close to 100% to each other. And yet, these three species are clearly distinguishable when you go by biochemical method of identification and DNA hybridization. So, are you getting the limitations of 16S rRNA? So, the relatedness by based on DNA homology is again less than 50%. And by biochemical methods, they show three different species which are not going to be showing any variation, considerable variation when you are doing 16S rRNA sequencing. So such examples, they indicate that small subunit sequences, that is 16S rRNA or 18S rRNA in eukaryotes, similarity, even though they are showing to a very high level, does not in each case imply identity or accuracy in microbial identification. Very important statement it is, that describes the limitation of 16SrRNA or 18SrRNA sequencing as a tool for taxonomic purpose. So many investigators have found resolution problems with the genus and that of the species also when it is 16SrRNA data is used. Now, for example, is family of Enterobacterias. So Enterobacter and Patonia. These are and the rapid growing mycobacteria. Then Acinetobacter. All these are showing some or the other problem when it comes to nomenclature and taxonomy using, taxonomy using 16SrRNA. A further problem regarding resolution of 16SrRNA gene sequencing is concerned with identity or very high similarity scores. So reports have documented 16SrRNA gene sequence similarities or identity for Streptococcus mitis and the other non-fermenters. Now see, Streptococcus mitis is showing similarity with non-fermenting bacteria when the method used is 16 when the method uses 16s rRNA sequencing. We know that Streptococcus mitis group and other non-fermenters, how they can be related to each other, right? So but such kind of similarities are shown when we go for 16s. Now the concern is 16S rRNA sequence data cannot provide a definitive answer to distinguish between recently diverse species. What are recently diverse species? Diverging from one point. So that selection is generally happening during evolution on account of various natural environmental circumstances. So through adaptations maybe, or through a gain of certain sequences through transformation, or like horizontal gene transfer processes, etc. So if the two species are recently diverse, then you won't be able to separate them on the basis of 16S rRNA, because 16S rRNA regions are, we are trying to match with those which are highly conserved regions, and there is variability only in specific regions, but that variability may not be analyzable when the species are diverged, if the species have diverged recently. Another concern when it comes to 16s rRNA is the difference between the closest and next closest match to the unknown strain is 0.5% divergence. That means 99.5% similarity. In such circumstances, small differences cannot justify choosing the closest match as a definitive identification, right? So when there are different organisms, they are sequenced and you are trying to find the closest match, because you are trying to classify them, you're constructing the cluster for the community, then the distance between the two species or two isolates that you have to measure. And when it is 99.5% or more similarity, then there is a challenge in finding out the closest match as a definitive identification, and there where is the limitation of 16SrRNA sequencing method, right? Now when it comes to 18SrRNA biodiversity studies, as we have seen, it is again a common molecular marker for biodiversity identification of eukaryotic isolates. Now the concern is, similar to 16SrRNA, 18SrRNA gene has 9 variable regions, right? And you have to go for 18S rRNA full length sequence or partial regions like V2, V4, and V9. So V9 has higher resolution at genus level, right? What is the important thing? When it comes to 18S rRNA sequence, V9 has a greater resolution for genus, and V4 is most important divergent region in the length, so would be a marker for a study of species, right? Here is one example of the species, but right, yeah. So V4 region is found to be very important for species level resolution. So let's see how these variable regions they look like and what are they called as. So ITS are internal transcribed spacer regions. They are located between 18S and 58S rRNA genes and have a high degree of sequence variations. Similar to 18S rRNA, ITS is often used in metagenomic analysis. So here you have sequences like 18S rRNA sequence, which is parallel to 16S in prokaryotes, and you have variable regions, right? V1, V2, V4, like till V9. That is one concern. And another is ITS regions, which are used for genomic analysis and clustering or taxonomic position of fungi and higher organisms. So here the ITS regions are used. They are used for metagenomic analysis. So when it comes to 18S rRNA, mainly used for resolving taxonomic studies. ITS region is mainly used for diversity studies, right? So uh, these are the schematic representations for, so 18S, 28S, and in between you have the two ITS regions, internal transcript spacer regions. So this is 18S, this is 28S, and these white blocks, they indicate ITS regions, right? So they are important when it comes to finding out the diversity of fungi or yeast, which are eukaryotic organisms. So ITS regions and 15S, 18S, 5.8S regions, 28S regions. So in between are interspersed these ITS regions, and each ITS region has its own cognizance or own importance in in finding out the diversity between the fungal isolates. Now, primers for 18S rRNA sequencing. There are two things. 18S rRNA sequencing is one, and second is ITS region profiling. We are talking now about primers for 18S. So, most common primers which uh, are there, which are NS1 and NS8. We can obtain a greater length than 1600 base pair. So the full length of 18S rRNA is approximately 1800 base pair. So the primers which are commonly used for amplification of the amplicon of 18S rRNA, yeah, these are a few of them: NS1, CNS1, NS2, NS3, up to NS8 you have. Alright, this is the TM value, melting temperature, for that sequence, and these are the primer sequences which are commonly used. This is the name of that sequence used. Of course, alternatively, you can go for the different databases which are available. For example, here one database, Silva, and another is UKREF. So EUK for eukaryotes, REF for reference, UKREF, which is going to help you in designing the primers. So very similar to 16S rRNA sequencing you are going to do. Now when it comes to 18S rRNA, it is very similar to 16S that of bacteria, and its gene sequences and associated transcript spatial regions, that is INTS regions, right, are used for classifying fungi and eukaryotes. Now we have seen in the previous slide what are the ITS regions. Now in this slide we are seeing the variability regions, right? So you have V1, V2, like that up to V9, that are the sequences. So if you look at the length of this region is 1 to 1542 base sequence, right? So of course you are going to go for sequencing with ITS regions or sequencing with 18S rRNA regions, and you can find, but co-sequencing of both, that is 18S and ITS, can provide more comprehensive classification of fungi. So these are the there where uh, it is telling you about the importance of both the regions, right? You cannot just rely on only ITS, or you cannot just rely on V1, V2. As in previous examples related to bacteria, we have seen how a specific region, say V3, is important, or how specific region V9 is important in specific type of species. It is not universal rule ki V1 for this or V2 only is good, not like that, right? So species wise it may vary. Now here is similar in the bacterial 16S rGNA. So you have rDNA repeats. The eukaryotic 18S rRNG has conserved and variable regions, and that is how the sequence would look like. And further fractionation is shown here like that. All right. So what you have is MTS1, so 5S, 18S, 5.8S, 25S, and 5S. So in between you have the regions which are ITS regions, right? And so the ITS regions and the hypervariable regions within the sequence, that is 18S के अंडर जो variable regions है वो ये है. And ITS regions देखो, these are the ITS regions, and when we are talking about 18S, 18S के अंदर जो variable regions है, they are shown like that, right? So they are important in classifying fungi. Now, based on development of sequencing technology, when we are using 16S or 18S or ITS RNA gene sequencing, it is best tool for study of bacterial and fungal taxonomy and molecular phylogeny. True, but we have seen what are the limitations and how you should go about when it comes to species level identification or clustering of the isolates. So yes, there are long read sequencing technologies like PacBio or SMART, right? CD Genomics, they can provide full length sequencing services to better help to your research. So it is just to make you aware of the terminologies, ki what are the different types of sequencing technologies that are used and what are the brands which are on the market and giving you good output. So full length sequencing, when it comes, the extraction of microbial DNA from a given sample to sequencing and sequence match we have seen, right? So here is the concern which is important is uh, the third generation full length sequence can provide more comprehensive and intricate strain level analysis, rendering the overall research outcomes more relevant. What does it mean? Third generation is Sanger's generation, Maxim Gilbert generation, and then you have next generations coming up for sequencing methods, right? Advancement. So इसमें से जो third level generation methods जो है, they would give a comprehensive and intricate strain level analysis. So it is species, genus, species, and strain, right? So uh, that is very important statement. And last statement is another important statement, that is the approach which includes multi-omics correlation and multi-omics, multiple omics, genomics, metabolomics, right? So multi-omics is going to be very important when you are going for clustering of organisms and their classification. So 16S, 18S, their limitations. In 18S, you have uh, uh, I mean in rRNA region of eukaryotes, you have ITS regions. So importance of ITS within 18S, hypervariable regions, importance of those, and then co-sequencing of both ITS and 18S regions, and importance of hypervariable regions in specific group of organism in narrowing down to the species level. And last point that we have talked here is strain level, that is third generation sequencing useful for. So the method of sequencing, right? Sequence that you are amplifying and sequencing out, how it is sequenced, which method you have used. Is it co-sequencing for both regions, ITS and the hypervariable regions? All these things are going to decide the outcome of your results. Is it going to be a true 100% complete classification that you are going to get, right? Now advantages ki against ITS region alone or against 18S region alone, parts variable region ke, if you go for full length 18S or complete ITS amplicon sequencing, what is the use? Of course, longer the reads, better is the accuracy, higher species identification and resolution and precision you can get.